Hi and Assalamualaikum. My name is Wansara Akila binti Warna Hussein, and today I would like to do the experiment, the rate of photosynthesis. So let's go.
The title of this experiment is the rate of photosynthesis. There are two objectives. First is to determine the effect of light intensity or the rate of photosynthesis, and the second to study the influence of sodium bicarbonate solution toward the light harvesting and energy breakdown in plants. Introduction. Photosynthesis is the process by plants and certain other organisms that will transform light energy into the chemical energy. To build simple sugars, photosynthetic organisms need water, light energy from the sun, and also carbon dioxide from the air. The equation of photosynthesis is 6 molecules of carbon dioxide react with 6 molecules of water and light energy to produce glucose and 6 molecules of oxygen. The sodium bicarbonate provides dissolved carbon dioxide. The solution provides water, and the light source provides light energy. The light-dependent reaction, which is the first stage of photosynthesis, it is to convert solar energy into chemical energy in the form of NADPH and ATP, which are used in the light-independent reaction and the full of assembly sugar molecules. There are two types of photosystems in the process of light-dependent reaction, which are photosystem 1 and photosystem 2 that are found in the thylakoid membrane inside the chloroplast. The reaction begins in PS2, and after that the election flow goes to cytochrome B6F and then to PS1. The net reaction of all light-dependent reaction in oxygen of photosynthesis is the effect of those steps is to convert light energy into chemical energy in the form of ATP and NADPH. Light-dependent reaction, or calling that Calvin cycle, is the reaction that uses energy stored by the light-dependent reaction to form glucose and other carbohydrates molecule. This process organized into three phases, which are fixation, reduction, and regeneration. Plants need both photosynthesis and respiration for their ability to function in the light. And in the first hypothesis is, as it rises from low light intensity to higher intensity, the rate of photosynthesis will increase. And the second is, the floating disk is better when it meets the water and sodium bicarbonate solution under table lab.
This is the sodium bicarbonate, 0.5 gram of sodium bicarbonate, and mix it with 50 ml of tap water. Okay, and then make a leaf disc, 10 leaf discs. And put the leaf disc into the syringe. 10 leaf discs into the syringe. 6 to 8 ml into the syringe. Okay. All the disc leaves are seen, and then we have to pour it into the glass.
The graph from my experiment. As you can see, leaf disc fluid in suspension about 3 cm is faster than suspension about 10 cm. Discussion. This is the answer from question 1 on how does the suction help the leaf disc to sink. This is the answer from question 2 on if you conduct the experiment without baking soda, what will happen to the leaf disc and why. This is the answer from question 3 on why do the leaf disc in the baking soda solution, which is treatment, begin to float. And this is the last answer from question 4 on what happened to the leaf disc in the dark. Justify your answer. In a conclusion, we know that the function of sodium bicarbonate solution in photosynthesis is to raise the rate of photosynthesis. The function of sodium bicarbonate in this experiment is to provide carbon dioxide for photosynthesis to occur. Once photosynthesis occurs, sugar is produced. The energy used for the light-independent reaction is ATP from light-dependent reaction and sugar energy. Next is about cellular respiration, which does not need sunlight or the source of light to make it happen. If there is no sunlight, the photosynthesis will occur like independent reaction, as known as Calvin cycle. In the dark, the leaf will sink, but some of the remains float. The photosynthesis will occur faster if the light intensity is higher.